Hello everyone. In our today's lecture of fermentation technology, we are going to start our next topic, which is downstreaming process. Downstreaming process refers to the recovery and purification of biosynthetic product, particularly pharmaceuticals, from the natural sources such as animals or plant tissues, or fermentation broth, including the recycling of salvageable components and the proper treatment of disposable waste. Generally, now in the fermentation technology or in the fermentation plant or in any industry, whenever the process is of product formation is completed, एखादा product completely बनवून झाल्यानंतर the next important step is your downstreaming process, which includes the recovery of your product, means the removal of your product from the fermentation plant. After removal, the purification of that product. After purification, the disposal or the reuse of the remaining material. All these processes comes under the downstreaming process. It is a very essential step in the manufacturing of different types of products like antibiotics, hormones, antibodies, different types of vaccines, as well as uh, different types of enzymes used for diagnostic purpose, and for the recovery of such products, the downstreaming process is very important. These are the general steps in the downstreaming purification. Whenever, now it completely depends whether your product is intracellular or extracellular. As we all know that in fermentation process we use different types of microbial strain, and these microbial strains are responsible in the conversion of your substrates into product. While performing this process, the microorganism have the ability to produce the desired product by two different means. Some bacteria or some microorganism can produce the product intracellularly, means inside the cell the product is produced, and by some organism extracellularly, means outside the cell the product is produced. Depending on the uh, production area of the product, it depends the which type of technique should be selected for the purification of the product. First, whenever the process is completed, the cells are separated from the fermentation broth. After the cell separation, if the product is intracellular, then the cell disruption is required, the breakdown of cell is required, and after the breakdown of cell, debris separation is carried out with the help of precipitation, and then the purification process is carried out. If product is extracellular, then directly solid liquid substances are separated, then they are concentrated, and further purification processes are carried out in the downstreaming process. Now there are various stages of the processing that occurs after the completion of the fermentation or bioconvergence stages, including separation, purification, packaging of the product. Means once the product is produced, after that few processes are carried out, which are separation of your product, after separation purification of that product, and after purification proper packaging of that product is carried out, which are included in your downstreaming process. Here are the stages in the downstreaming process. First one is removal of insolubles. Here from the fermentation broth, you separate all the cells as well as the cell debris and your product from this soluble matter. You separate your product, means isolation of product is carried out. After isolation or the separation of product, purification is carried out, and then the product polishing and packaging is done. In few cases or in the few product production, the recovery methods, some product can be separated by single recovery method, and for this few product they require combination of two or more stages for the separation and purification of the product. We'll see the stages one by one. First one is removal of insolubles. Here the separation of cells, cell debris and other particle matters is carried out. Now as we all know that in the fermentation or in the fermenter, we add our nutrient broth, then the inoculum is added. Afterwards the temperature and pH is adjusted, aeration is carried out, and then the further process of fermentation is carried out in the fermenter. Means after the completion of whole process, there are lots of remaining cells as well as cell debris and the other particular matters are present अदर दॅन प्रोडक्ट सोबत हे सगळे मॅटर्स पण काय असतात त्या फर्मेंटरमध्ये प्रेझेंट असतात. मग पहिली स्टेप आपली काय असते की हे जे काही सेल्स असतील किंवा सेल डेब्रिज असतील किंवा बाकीचे जे काही पार्टिकल्स असतील ते सेपरेट करायचे. अँड दिस कॅन बी अचीव्ड बाय फिल्ट्रेशन, simple centrifugation or sedimentation, even floculation. Floculation is a process where a solute comes out of solution in the form of floc or flakes. Here the different types of agents are added so that floc formation will take place, and in this floc जसं एक फेस तयार होतो आणि या फ्लॉक्समध्ये तुमचं जे काही सॉलिड पार्टिकल्स असतील किंवा सेल डेब्रिज असतील ते सेल डेब्रिज आपण इझिली काय करू शकतो, रिमूव्ह करू शकतो, किंवा सिम्पली फर्मेंटरमधलं सगळं सबस्टन्स जे आहे ते ॲज इट इज स्टेडी कंडिशनला ठेवायचं. ड्यू टू ग्रॅव्हिटेशनल फोर्स ऑल द पार्टिकल्स गेट सेटल्ड ॲट द बॉटम ऑफ द कंटेनर्स, and in this way all the insoluble debris or matter can be separated from the product. Next step is your filtration. Now the filtration is a mechanical operation used for the separation of solid from the fluids. Here your fluid is your broth and solid matters are your cells, microbial concentration or the cell debris, and it may also contain your product. So simply by carried out filtration process we can separate these solid particles from the fluid by interposing a medium to porous membrane through which fluid can pass but the solid in the fluid are retained. Now when we are using filters, all the liquid content are passed through the filters and only the solid content whichever are present in your fermentation broth remain on the filters, and these solid particles get deposited on the filter and forms a layer which is known as filter cake. And this cake grows at the rate at which particles are brought to the surface. Means the amount of as the amount of particle increases, the amount of cake formation or the rate of cake formation also increases, and all the fluids is passed through these cakes and the filter medium is obtained. Now, in this diagram, the continuous rotary filter is shown. Now, this is one type of filter which is used for the separation of solid particles from the liquid broth. It is one of the most commonly used type of filter in the fermentation. The drum is pre-coated prior to the filters. The small agent of coagulating is added before the to the broth before it is pumped into the filter. The drum rotates under the vacuum and the thin layer of the cell sticks to the drum. The thickness of the layer increases in the section designed for forming the cake. Now, here we can see this is your fermenter. Through this fermenter, here when the fermentation process is completed, all the liquid matter or the broth is collected inside this drum in the कंटेनर. या कंटेनरमध्ये काय केलं जातं, फर्मेंटरमधनं येणार जेवढा काही ब्रॉथ असेल तो कलेक्ट केला जातो. अ रोटरी ड्रम इज सेट ऑन दॅट कंटेनर. हे जे व्हील लाईक स्ट्रक्चर तुम्हाला जे दिसतंय, दिस व्हील लाईक स्ट्रक्चर इज युअर रोटरी ड्रम विच इज परफोरेटेड. ह्या ड्रमवरती किंवा या व्हील लाईक स्ट्रक्चर्सवरती स्मॉल पोर्स प्रेझेंट असतात आणि ह्या पोर्समधनं तुमचं लिक्विड जे आहे ते फिल्टर होत असतं. ॲट द सेंटर द व्हॅक्यूम इज सेट सो दॅट तुमचे जे काही सॉलिड मॅटर असतील ते सॉलिड मॅटर काय होतील, त्या रोटरी ड्रमच्या किंवा त्या सायक्लिक व्हीलच्या सरफेसवरती अटॅच राहतील. व सरफेसवरती अटॅच राहण्यासाठी काय केलं जातं, ब्रॉथमध्ये कोॲग्युलंट्स ॲड केले जातात सो दॅट ऑल द सॉलिड मॅटर विल गेट कोॲग्युलेटेड. म्हणजे एका जागी ते काय होतील, जमा होतील. वन्स द प्रोसेस इज स्टार्टेड. मग प्रोसेस स्टार्ट कशी होते की एकदा तुम्ही ड्रममध्ये ह्या कंटेनरमध्ये ज्यावेळेस तुम्ही ब्रॉथ ॲड करतात, ही व्हील किंवा रोटरी फिल्टर जे आहेत ते कम्प्लिटली सर्क्युलर मोशनमध्ये मूव्ह व्हायला सुरुवात होते. ॲज इट मूव्स इन सर्क्युलर मोशन, all the solid particles get attached to the pores of the rotary drum. त्या रोटरी ड्रमच्या किंवा रोटरी फिल्टरच्या पोर्सवरती काय होतं, सॉलिड पार्टिकल्स जमा होऊन तिथे काय होतं, लेअर तयार व्हायला सुरुवात होतं. आणि जे काही तुमचं रिमेनिंग फ्लुइड असेल ते रिमेनिंग फ्लुइड कलेक्ट केलं जातं in separate container, and by this way the layer of the matter gets developing on the particular areas of the filter. त्या फिल्टरच्या पर्टिक्युलर एरियाजमध्ये सॉलिड मॅटरचं काय होतं, एक लेअर तयार व्हायला सुरुवात होते आणि तुमचं जे लिक्विड कंटेंट असेल किंवा जे फ्लुइड असेल ते इझिली काय करता येतं, तुम्हाला सेपरेट करता येतं. There are few points that should be considered while selecting the filter medium. It should have the ability to build the solid. सॉलिड्सला जमा करण्याचं किंवा गॅदर करण्याची ॲबिलिटी त्या फिल्टर्समध्ये प्रेझेंट पाहिजे. इट शुड मिनिमम रेजिस्टंट टू द फ्लो द filtrate. Means the filtrate should be slightly viscous so that the solid it will move slowly through the filters and the solid matters will remain or retained on the filter. They should be resistant to the chemical attack. This filter should be of minimum cost and long life. This was all about the filtration technique. There are few more types of filters which are used for the separation of the solids from the liquid or your fluids. Next step is centrifugation. Now centrifugation process is generally used for the separation of particles having size 100 to zero point 0.1 micrometer by using the gravitational force. Now here the gravitational force is used or applied on the liquids so that the solid matters will get easily separated. It depends on the particle size, density difference between the cells and the broth viscosity, means the separation of your product and the other matter. It completely depends on the particle size, means the size of the particle, as well as the density difference between the cells and the broth viscosity. Here for the separation purpose the centrifugal force is used. The different types of centrifuges are used for the separation depending on the type of the product. आता प्रोडक्टचा टाईप कसं आहे किंवा ते प्रोडक्ट कुठे प्रोड्यूस झालं याच्यानुसार आपण काय करतो, सेंट्रिफ्युज कुठलं सिलेक्ट करायचं हे त्यावरती कम्प्लिटली डिपेंड करतं. Then the more dense component migrates away from the axis of the centrifuge, while the less dense particle migrate towards the axis. There are different types of centrifuges used for the separation of the particles, such as tubular bowl centrifuge, which has the structure just like a tube. Then the multi-chamber centrifuge are used. Disc bowl centrifuge are also used in the separation of the product. Then the next type is your sedimentation. Now the sedimentation process is carried out for the larger particles or the particles having the size range more than 100 micrometer. Now generally for the process sedimentation process, it requires more time because we have to add all our broth or the medium inside the container and left it for few hours so that all the solid matter present inside the broth will settle at the bottom. And these process is generally used in the waste disposal process or in the treatment of effluent. The sedimentation process is used, and it is completely dependent on the gravitational force or the gravity. Then the next type is your floculation, means floc formation. Now it is the process where a solute comes out of solution in the form of flocks or flakes. आता इथे काय होतं की इन द फॉर्म ऑफ फ्लॉक्स तुमचं जे काही सोल्यूट असेल किंवा जे काही सॉलिड मॅटर ब्रॉथमध्ये प्रेझेंट असेल ते त्या सोल्युशनमधनं बाहेर येतं. म्हणजे डिफरंट टाइप्स ऑफ flocculation agent or flocculating agents are added inside the broth, and or certain aeration and agitation is provided, so the floc formation takes place inside the broth and the solid material present inside the medium will get separated in the flocks. Now generally the finer particles of 0.1 micrometer in water remain continuously in motion due to electrostatic charges, which cause them to repel from each other. Once their electrostatic charges is neutralized, the finer particles start to collide and combine together. Now, in whenever the finer particles are present inside the water of less than zero point 0.1 micrometer size, they due to electrostatic force they continuously keep on moving. Once these forces change or neutralize, all these smaller finer particles starts colliding with each other, and a larger or heavier particle is formed, which is known as floc. Next is your product isolation, means separation of your product. Removal of those components whose properties vary markedly from that of the desired product. Product isolation means the separation of the components which has the different market value, like what, like for example water is a chief impurity. In this isolation steps are designed to remove it. First step is to design the method by which the product can be isolated. Then reducing the volume. Here why there is need of reducing the value, so that in less volume more product can be easily isolated. Then concentrating the product means to increase the concentration of product and removal of waste material. In this process, different for the product isolation, different types of processes can be used. For example, liquid extraction, adsorption, ultrafiltration, and precipitation are the sum of the unit operations involved in the product isolation. Means these different types of processes can be used for the isolation or the separation of the product. All these different processes like liquid extraction, adsorption, ultrafiltration, precipitation, all these process we will study in detail in our next lecture.